Hello students, this is Dr. Anjani, Assistant Professor in Biotechnology. Welcome to e-learning sessions in genetics. In this session, we learn about Mendel's laws of inheritance. Between 1856 and 1863, Gregor Mendel, scientist named Gregor Mendel, he is considered as father of genetics, conducted the hybridization experiments on garden pea. For this, in the garden pea, for conducting the hybridization experiment, he selected the garden pea because of various important characteristics. He selected the pea plant for his experiments because it can be easily grown and maintained. They are not naturally self-pollinating but can also be cross-pollinated. So they are self-pollinating, but if you want to bring some new character in the pea plant, you can also go for the cross-pollination. And it is an annual plant, short-lived. Therefore, many generations can be studied within a short period of time. So you can repeat this experiment, or he repeated this experiment for various characters and he ended up with the same results. So the results were reproducible when he conducted the repeated experiment. And this plant has several contrasting character. For example, if you consider one phenotype, height is the phenotype of the plant. Height is the phenotype of the plant. So here it has the two contrasting phenotypic character. When you consider this is the height of the plant, so it has two contrasting character like tallness, and another one is short, or it is considered as dwarfness. So these characters are very useful in differentiating the two alternative expression of the same characteristic, or two alternative expression of the same particular phenotype. So each contrasting character, he had many contrasting character and he studied this experiment, hybridization experiment, and repeatedly he could get the same result. Based on this, so here he could do the hybridization experiment with the seven different contrasting character. For example, in the seed, here in the seed, when we observe the seed pea plant, we will find the rounded seeds as well as wrinkled seeds. That is one character. So seed form, when you consider, round and wrinkle are opposite alternative, or the contrasting characteristic of the same character, that is the seed form, and also gray and white. So these two are the contrasting characters of the seed form. Similarly, cotyledons, when you take, so cotyledons were yellow and green. So the cotyledon color was having the two contrasting character, could be easily differentiated. And even in the flower, it had white flower and the violet flower. So the flower color also had two alternative expression. One is white and another one is violet. Similarly, the pod form. So the pod, some pods were full and some were constricted. So this pod form also had the two contrasting character. And then the pod color. When you consider the pod color, it had the two contrasting character, that is the yellow and green. So the yellow and green are the alternative expression of the pod color. That is, the pod color is the character. And when you take the stem, you may have the axial inflorescence, axial pods and flowers along, or the axial inflorescence, or the terminal inflorescence with the flower tops. So these two are the characteristic where the plant bear the inflorescence. It may be axial or the terminal. Similarly, when you take the stem size, they may be long or they may be short. So that is the height of the stem. So height of the plant, when you consider, it may be tall or it may be short. So short plants are considered as the dwarf plants. They are considered as dwarf plants. Like this, he studied 7 different characteristics in pea plant when he did the hybridization experiment. After studying the hybridization experiment, he could able to the postulate few laws, and he designated the experiment what he conducted as monohybrid cross experiment and dihybrid cross experiment. Monohybrid cross is a cross between the two parents differing in single contrasting character. It may be height, it may be the pod color, it may be the pod form, or anything. So, such type of experiment he called as monohybrid cross. Similarly, when a cross occurs between the male parent and the female parent, and they are differing in two contrasting character. So here it may be the height or the pod color. Okay, but here in the dihybrid cross this involves two contrasting character. It may be the pod color, it may be the pod color and the seed form. Two characteristics were considered, or it may be the seed color and seed form. So seed color when you take it will be yellow and the green, and the seed form when you take it is round or it may be constricted, that will be, or it is wrinkled, so that may be represented by smaller. So it's a cross between the two parents differing in two contrasting character, it is called dihybrid cross. If a cross between the two parents differing in the single contrasting character, it is called monohybrid cross. So when he conducted these two experiments by repeated repetitions with various other contrasting characters, he could able to postulate three important laws. They are named as Mendel's laws, or otherwise independently they are considered as laws of inheritance, that will be studied under law of dominance, law of segregation, and law of independent assortment. After, when he completed the experiments with the monohybrid cross and the dihybrid cross, he could able to postulate the few laws. They are called laws of inheritance or Mendel's laws of inheritance, and will be studied, or he has postulated law of dominance, law of segregation, and law of independent assortment. Now we'll see these laws individually. First, we'll take up law of dominance. This is also called Mendel's first law of inheritance. According to this law of dominance, according to this law of dominance, in a hybrid offspring, in a hybrid offspring, only dominant trait will be expressed. Dominant trait in the phenotype will be expressed, but not the recessive trait. So hybrid means, if you just consider the height of the plant, one factor is the dominant trait and another one is the recessive one. So the dominant trait here is the tallness. It is tallness, and the recessive trait here is dwarfness or the shortness of the plant. When in a hybrid, when the both the factors are heterozygous, that means one is for the tallness and another one is for the dwarfness. So these two are the alternative expression of the one character, that is height, that is height. So when you consider the height of the plant, some plants are tall, some plants are dwarf. So these two are the alternative expressions, and tallness is controlled by capital T and dwarfness is controlled by small t. In a hybrid, when pair of factors are brought together, when pair of alleles are brought together, only the dominant factor expresses its phenotype, and all the plants will be tall, whereas the factor or the allele whose phenotype is not expressed is considered as the recessive trait. Is considered as recessive trait. So that is here the dwarfness. So this is called law of dominance. In a hybrid offspring, when you have the heterozygous condition for the particular character, heterozygous condition for the particular character, usually the dominant trait expresses its phenotype, whereas recessive trait fails to express its phenotype. This is called law of dominance. This is called law of dominance, and the phenotype which is expressed is called dominant trait, and the phenotype which is not expressed in the hybrid, it is called the recessive trait. So this is regarding law of dominance. Next, we'll move on to law of segregation. The law of segregation states that when pair of factors brought together in a hybrid, when pair of factors brought together in a hybrid during the production of the gametes, during the production of the gametes, two copies of each hereditary factor, two copies or the two alleles of the particular character segregate during the gamete formation so that offspring acquire one factor from each parent. So that offspring acquire one factor from each parent. In other words, allele, alternative form of the gene, alleles, pair of alleles segregate during the gamete formation and reunite randomly during fertilization. Randomly they reunite and they result in the next generation. That means when pair of factors brought together in a hybrid, during gamete formation, each factor segregate independent of other. Each factor segregate and hence you will get the single factor for the gamete. We know gametes are haploid. Gametes are haploid. Parents are diploid. So they have the 2n or you have the 2 capital T in case of the homozygous parent. So like small tt, that is the recessive parent. So in the F1 generation, when there is formation of the hybrid, you will get the hybrid like capital T and small t. So this hybrid during gamete formation, during gamete formation, 50% of the gamete receives the capital T. 50% of the gametes receives capital T, whereas 50% of the gametes receives the small t. So they segregate during the gamete formation and the each factor is pure for the gamete. It is pure for the gamete. Hence this law of segregation is also called as law of purity of gametes. It is also called law of purity of gametes because during gamete formation there is segregation of the factors, there is segregation of the factors and each gamete receives the single allele or it receives single factor. This is called law of segregation. Next, we'll move on to the law of independent assortment. Law of independent assortment is also known as Mendel's second law of inheritance. It's also known as Mendel's second law of inheritance. The law of independent assortment says that when cross occurs between the parents differing in two or more contrasting characters. So it's a law which will explain the inheritance of the characters in a hybrid where the two parents are differing in two contrasting characters or more than two contrasting characters. So it may be yellow seed color, round seed color or seed color as well as the seed texture. So both the characters are considered. For each character you have the alternative expression. So totally in case of the dihybrid cross, you will have the four alternative expression: yellow and round, green and wrinkled. So these four expressions will be observed. So the two parents differing in two or more contrasting characters, factors segregates during the gamete formation independent of one another, independent of one another, and hence in the F2 generation, that is the second filial generation, we will find the new combinations or different new traits are expressed in the F2 generation. This is the law of independent assortment. So these laws will be asked for short notes in your examination. They will ask you the question like this. They state the law of dominance, state the law of independent assortment, state the law of segregation. So that time you have to write these laws. What this law of independent assortment says? It is the law of independent assortment says that when cross occurs between the parents differing in two or more contrasting character during gamete formation, there is assortment of the character. So they segregate independent of one another during the gamete formation, and hence we find the new different traits in the F2 generation. This is called law of independent assortment. Now we'll take up monohybrid cross, where it explains the law of segregation as well as law of purity of gametes. In monohybrid cross, Mendel took two pea plants of opposite traits, or he took the one male plant and the one female plant. They are differing in single contrasting character. Single contrasting character. So in his experiment, he took one short and one tall plant and he crossed them. He found in the first generation offsprings were tall. The offsprings were tall. So its first generation is called as first filial generation or the F1 progeny or F1 generation. When he crossed, hybridized one tall pea plant with one short pea plant, in the first generation, the first offsprings, all of them were tall plants. All of them were tall plant. So this explains the law of dominance. This explains the law of dominance. When pair of factors brought together in a hybrid, usually dominant allele or the dominant factor expresses its phenotype. So in the F1 progeny, all the plants or all the offsprings were tall. Then he crossed F1 progeny obtained were self. So F1 plants were self crossed. When he did this experiment, he obtained both tall and the short plants in the ratio of 3 is to 1 in F2 generation. In F2 generation, when he did the selfing of the F1 plants, F1 progeny, F1 male plants were crossed with F1 female plant, he got the next generation that he referred it as F2 generation. And in the F2 generation, he got both the tall plants and the dwarf plant. The tall plants number were 3 and the tall plant number were 3 and the dwarf plant numbers were only 1. But when he selected the F1 progeny for the self hybridization, selfing, all the F1 plants were tall. All the F1 plants were tall. But in F2 generation, second filial generation, he got both the tall as well as short plants, otherwise referred as the dwarf plants. So this dwarfness was hidden in the F1 generation in the hybrid without showing its phenotype. Without showing its phenotype. So this is called as monohybrid cross. Mendel even conducted this experiment with other contrasting traits like green peas versus yellow peas and round versus wrinkled, etc. In all these contrasting character inheritance, he could get only 3 is to 1 ratio in the F2 generation, and all the F1 plants were dominant trait containing plants. They were following the law of dominance. And in the F1 generation, all the plants were tall. But in case of the F2 generation, he got both the dominant character containing plants as well as the recessive character containing plants. Let's see how he mathematically proved this experiment. He took the homozygous tall plant. It is called homozygous because it is having same type of the allele. So both the factors will code for the dominant character. Both the factor will code for the tallness of the plant. Was crossed with dwarf plant or the short plant, which is the recessive character. This is also homozygous plant, homozygous dwarf plant. Here, parents, he took homozygous tall plants were pea plants were crossed with homozygous dwarf pea plants. During gamete formation, since there is reductional division, all the gametes will receive only one trait or will receive only the 50% of the factor. So, all the gametes contained only the capital T. Whereas in case of the dwarf plant, all the gametes had only small t. So during fertilization, the was tall. Next plant was tall, but it had the factor of dominant uh, trait as well as the recessive trait. So this is called as a hybrid. So this is called heterozygous hybrid, heterozygous tall hybrid. And this generation, first generation is called as F1 generation, which had the tallness controlling or the dominant gene as well as the recessive gene. So this is called a hybrid. Now when he did the F1 selfing, when F1 plants were selfed, when F1 male plants were crossed with the F1 female plant, F1 female plant. So the F1 male plant genotype is capital T small t. F1 female plant genotype is capital T small t. During gamete formation, 50% of the gametes received capital T and 50% of the gametes received the small t in both the condition, both in male as well as in female. And he represented the fusion of the gametes using the Punnett square or the checker board. So this is called checker board. So was referred by the scientist Punnett. He represented the male gametes on the right side or the top of the Punnett square and the female gametes on the downward end of the square. So the male gamete produces capital T and the small t, and the female gamete produces capital T and small t. So during fertilization, when the fusion of the gamete takes place, this female capital T may fuses with the male capital T and results in the homozygous capital TT, that is homozygous tall plant. Similarly, the female capital T may fuses with the recessive trait of the male gamete and results in capital T small t. So here the factors are not same. That is called as the heterozygous tall. So this is heterozygous tall because this dominant allele or the factor will express its phenotype. Hence all the plants will be tall. So you got one homozygous stall and one heterozygous stall when the female capital T fuses with the gametes of the male plant. Now we'll take up the female recessive factor, that is small t, when it fuses with the capital T of the male gamete. It again results in heterozygous tall plant having the one capital T and another one is small t. Similarly, when this small t of female gamete fuses with the small t of the male gamete, it results in homozygous small tt condition. Homozygous small tt condition. Here there is no dominant factor or the allele and hence you will get the homozygous dwarf, or it is the dwarf plant because there is no capital T representation. That's why it is considered as homozygous dwarf. Now, when you see the phenotype, the external appearance of the four types of the plants we get in selfing of the F1 plant in monohybrid cross, we will get three will be tall, 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 tall. So three are tall, and one is dwarf. So the phenotype of the F1 monohybrid cross is the 3 tall, 1 dwarf flat, and this ratio is called as the phenotypic ratio. What is this phenotype? It is the process by which we define the phenotypic appearance, or the external appearance of the organism is called as the phenotype. So morphological appearance of the phenotype, whether it is tall or whether it is dwarf, or whether it is tall or whether it is yellow or whether it is green. So externally how it looks, that is called the phenotype of an organism. So the monohybrid phenotypic ratio is 3 is to 1. And at the same time, when you consider the genotype of the organism, the genetic composition of the each plant, that is the representation of the genetic composition of an organism is called genotype. So when you observe the genotypic ratio, one is homozygous tall and two are heterozygous tall, and one more is the homozygous dwarf. So one is homozygous having capital TT and two are having capital T and small t, and again one is the having both the factors are dwarf, or both the factors are the recessive factor. So that's why this is called genotypic ratio. So this will be asked in the examination. Write the phenotypic ratio of the monohybrid cross. It is the appearance, the phenotypic appearance of the organism is called the phenotype. So the ratio which tells you how many are tall, how many are dwarf, it is called phenotypic ratio. Similarly, when you see the genotype of the organism, genetic composition of the organism, whether it carries both the dominant alleles or whether it carries the heterozygous allele, then it is called as the genotype of the organism. The ratio which tells you the genotype of the organism is called as the genotypic ratio. So this is regarding the monohybrid cross experiment the Gregor Mendel conducted by taking the height of the plant. Now we'll move on to monohybrid backcross. What is meant by backcross? Backcross is the crossing or the hybridization of the F1 plant with one of the parent. It may be the dominant parent or it may be with the recessive parent. That is called backcross. What is backcross? Backcross is the crossing of the F1 plants or the F1 offsprings with a dominant parent or the recessive parent. It is called the backcross. If you cross the F1 plant with the dominant parent, it is called as the dominant backcross. If you cross the F1 plant with the recessive parent, it is called as test cross. It is also called as the recessive backcross, or it is also called as test cross. This test cross is very important because this helps us to know the genotype of the organism, whether the parent has the homozygous condition or the heterozygous condition could be identified by using this recessive backcross. It is otherwise called as test cross. First, we'll take up the dominant backcross. It is a cross between F1 parent with a dominant plant. Now, the F1 plant will produce two types of the gametes, that is capital T and small t. But the dominant parent, 100%, all the gametes will be having only one type of the genotype. All will be having the dominant factor. When this is crossed, when the fusion of the gamete takes place, it results in all the plants are tall. All plants are tall. All are carrying the dominant character because they have either dominant heterozygous condition or the dominant homozygous condition. This is dominant homozygous condition, and here you have the dominant heterozygous condition, due to which you will have all the plants will be tall. So it is 100% tall. Here you cannot define whether the parental plant is having the homozygous condition or the heterozygous condition. But if you want to know the given plant is having the genotype homozygous or heterozygous, you cross this F1 plant with the recessive parent. This is called test cross. So here we'll see when F1 plants are crossed with the recessive parent, F1 hybrid produces two types of the gametes. 50% will be having the capital T, another 50% will be having the small t. Another 50% will be having the small t. But the recessive parent will produce only one type of the gamete. All the gametes will be having the small t. All the gametes will be having the small t. So you represent that on the Punnett square. You represent that on the Punnett square. So there is fusion of the male and female gametes, and that results in the 50% tall plant and 50% dwarf plant. 50% will be tall plant and another 50% will be dwarf plant. So if you consider the proportion of the tall and the dwarf plant, it is 1:1. It is 1:1. So what is the monohybrid test cross ratio? It is 1:1. So this is frequently asked short notes question in the examination. Next, so this is represented here, test cross, monohybrid test cross has been represented. When F1 hybrid is backcrossed with pure recessive, homozygous recessive parent, so the law of segregation will result in 1:1 ratio. That means the 50% of the plants will be tall and 50% will be dwarf. 50% will be tall and 50% will be dwarf. So what is the monohybrid test cross ratio? It is 1:1. It is 1:1. So I think you understood the law of segregation or law of purity of gamete or Mendel's monohybrid cross experiments. Now we will move on to the Mendel's law of inheritance, where he has considered two contrasting character. Where he has considered two contrasting character. Here he has taken the seed color and seed texture. Seed color and seed texture. Seed texture means seed form, how it looks. So one is color and another one is seed form. So in the dihybrid cross is a cross between the two parents differing in two contrasting character. When a homozygous yellow round seed bearing plant was crossed with green wrinkled seed bearing plant. Green wrinkled seed bearing plant. All the F1 plants were yellow and round. All the F1 plants were yellow and round. But when F1 plants were selfed, the law of independent assortment takes place and it results in 16 different types of plants, 16 plants, total 16 plants. Among them, apart from the parental phenotype, yellow, round and green wrinkle, he also got the new combinations like the yellow wrinkle, and green round, plants in the ratio of 9 is to 3 is to 3 is to 1. Here the parental combinations are yellow round. Yellow round are totally are 9. Yellow round are totally 9. And the another parental combination is green wrinkled. Green wrinkled is only 1. So 9 are the dominant parental combination, that is yellow round, and 1 is the recessive parental combination, that is green wrinkled. But apart from this, due to law of independent assortment, he got 3 new combination, that is the yellow wrinkle and yellow wrinkle, and another one is the green round. So three plants were green round, three plants were yellow wrinkle. So these two are the new combination which were not there in the parental combination. So this inheritance is called as the dihybrid cross inheritance, or this will witness the law of independent assortment. In law of independent assortment, we take the two parental plants and the dihybrid cross parental plant differing in two contrasting character. When they were selfed, when they were crossed, you will get the F1 generation. All of them will be yellow round. All of them will be yellow round. But when the F1 plants were selfed, we get the F2 generation. We get F2 generation. In F2 generation, we get the 16 plants. We get 16 plants. Among them, 9 are yellow round, 1 is green wrinkled, parental combination. Apart from that, we also had 3 green round and 3 yellow wrinkled. These two phenotypes were not there in the parental combination. This is regarding the dihybrid cross. Next we'll see the dihybrid test cross or the back cross. So we know back cross is the crossing of the F1 parent with the either dominant parent or the recessive parent. That is called back cross. If you say it is test cross, it is the crossing of F1 parent with the recessive parent, then it is called test cross. So here F1 dihybrid plant, F1 dihybrid plant, here it has the genotype of the capital Y, small y, capital R, smaller, capital Y, small y, capital R, smaller. So YY represents the yellow color, RR represents the form of the seed, that is the roundness. But here the small YY codes for the green color of the seed, and the small RR refers to the wrinkled nature of the seed. So when these two were crossed, you will get the F1 generation. They will be heterozygous dominant having the capital Y and small y, hence it is yellow, and it has the capital R and small r, heterozygous, the form of the seed. So it is the again round because capital R is there. When this F1 plant is crossed with a recessive parent, recessive parent is double homozygous, will be having the small YY and small RR. So both the color as well as the texture of the seed controlling genes are in the homozygous recessive condition. They are the recessive alleles. When this was crossed with the F1 plant, so this F1 dihybrid, it is a double heterozygous condition both for the color as well as the texture. It results in the formation of 4 different types of the gametes. So here the law of independent assortment takes place. Here the factors assort independent of one another. Y can fuse with capital R, Y can fuse with small r, small y can fuse with capital R, small y can fuse with small r. It results in the randomization or the random mixing of the gametes takes place, or the alleles takes place, and results in 4 different types of the gametes, 4 different types of the gametes, and hence you will get 4 gametes on the right side or the top end of the Punnett square. Whereas the female plant, which is double homozygous recessive parent, code for the green and the wrinkled texture, will produce only one type of the gametes. So 100% all the gametes will receive only one type of the genotype, that is small y, small r. So in the test cross, when the fusion of the gamete takes place, 25% of the plants will be yellow round, 25% will be yellow round. They will be having double heterozygous condition for both the color as well as the texture. And 25% will be yellow wrinkled, which is the new combination, yellow wrinkled, which is new combination which is heterozygous dominant for the color and homozygous recessive for the texture, and hence it is yellow wrinkled. And the 25% will be green and round, which is having the homozygous condition for the color and heterozygous dominant condition for the form of the same, and hence again you'll get the 25%. And in case of the green wrinkled, it is again 25%. So 25% of the dihybrid test cross individual will be having the parental combination, that is the recessive combination, recessive phenotype, that is green wrinkled. So it is double recessive homozygous, double recessive homozygous. So dihybrid test cross ratio is 1 is to 1 is to 1. But of monohybrid test cross ratio is 1 is to 1. 1 is to 1. So, in this session, you learned about the Mendel's experiment by taking the pea plant and the few characters which are advantageous by selecting the pea plant, and how he formulated the three laws: law of dominance, law of purity of gametes, and law of independent assortment. And you learnt about monohybrid cross and then dihybrid cross, and in addition you learnt about monohybrid test cross and dihybrid test cross. I hope you understood this session. Thank you.